So what is an antigen? What is an antigen? We talk about an antigen being presented or so, and so on. Basically, again, as I mentioned, your body, as it looks at foreign molecules, it look at them as a shape. And these basically antigens, they can be either proteins, they can be polysaccharides, they can be lipids, they can be nucleic acids. So anything that is that has a shape, that has a molecular pattern on a surface, it is recognized. And usually, in most cases, when we have strong antigens, and usually those are proteins, usually they're going to be there are going to be certain peptides on their surface, and those are going to be the epitopes. Those are going to be the epitopes that is that your immune cell is going to respond to. So an epitope is a determinant that is on the surface of that antigen that interact with the immune cells, that engages the immune cells. And an antigen, depending on how big this antigen is, there are going to be a lot of these points on its surface. They're not necessarily identical. However, in the case of polysaccharides, they are identical. And that becomes very important when it comes to activation of B cells. So when an antigen is able to elicit an immune response, we call it an immunogen. So an immunogen is an antigen that can elicit an immune response, activate an immune cell, whereas an antigen is just something that is going to engage the immune cell. So all immunogens are considered antigens. However, not every antigen is an immunogen. So why is it not every antigen as an immunogen? Because for an antigen to elicit an immune response, it needs certain properties. It needs to be big. It has to have enough size in order to be recognized and be able to organize, to activate that immune cell. Their structure has to be complex. Their structure has to be complex enough. And it also has to be accessible. If your immune cell cannot reach that antigen, well, it cannot, cause an immune response to it. So it has to be soluble, right? Now, usually, if you look at this slide, I have put basically all these antigens in order of their immunogenicity increasing. So your proteins usually are the most immunogenic because they're big, they're complex, and they have lots of areas on their surface where there are many epitopes here, 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 here. And all of these are areas where your immune cells can interact. Your polysaccharides, your carbohydrates, they're also good immunogens. They're not as strong as proteins. And there will be lots of identical epitopes on their surface, and that comes in handy later. Now, as you go down the list, you get to lipids and nucleic acids and all of that, and haptens. Then the order of immunogenicity decrease. And these items here are usually antigenic, meaning they will respond, they will interact with immune cells, but their interaction is not very strong, and they are unable to, on their own, elicit an immune response. And therefore, usually, they're conjugated, they're coupled to a bigger molecule in order to elicit an immune response. And that comes in very handy when we're designing vaccines. A lot of vaccines we make are basically unable to elicit an immune response, like the Haemophilus influenzae vaccine. On its own, it doesn't. It's a hapten. But what we do is we conjugate it to a protein, and then it is suddenly able to elicit an immune response, right? So this is called conjugating a hapten to a bigger molecule, or coupling that a small molecule to a bigger molecule. So a hapten, basically, this is a small molecule. It has a very low molecular weight, and on its own, it cannot elicit an immune response. We add it to something else, and it becomes able to elicit immune response. So it is antigenic. It will bind an immune cell, but it will not activate that immune cell alone. An epitope is also known as an antigenic determinant, and it is that molecule that it's on the surface of that antigen that is going to interact specifically with that immune receptor. And when I talk about immune receptors, we have lots of immune receptors. And I'm going to go over those. I'm going to spend some time actually over these immune receptors. Now, there are different types of antigens when it comes to lymphocytes. There are T-independent antigens and there are T-dependent antigens. And usually, your T-dependent antigens are these antigens that are taken up by an antigen-presenting cell. They're digested, they are presented to T helper cells, and then these T helper cells will interact with the B cell. So the antigen will interact with an antigen-presenting cell. And then it will present it to a T cell, and that T cell will interact with the B cell and tell the B cell, you know what? I want you to basically become a better B cell, make better immunoglobulins, class switch. Don't make only IgM, make IgG or make something. So that can only happen in the case of T-dependent proteins. And these are basically protein antigens and T-dependent antigens, I'm sorry. And these are usually proteins. And where do these interactions happen? Usually these interaction happens in the follicular areas of the secondary lymphoid organs, in the follicular areas of the secondary lymphoid organs. Now, B cells can be activated by something other than a protein. They can be activated by polysaccharides. And as I mentioned, polysaccharides, they are these long molecules, and they have these multiple antigenic determinants on their surface, multiple epitopes on their surface, but they're all the same. And they will interact with many, many immunoglobulins at the same time. And they will elicit a signal that is strong enough to activate that B cell on its own without the help of a T cell. Now, the only problem with this picture becomes, do you know what the problem with this process here? Well, the problem with this, the cell get activated and the B cell will basically make more B cells. However, that B cell will not be able to change its class. It will not class switch because unless you have a signal from a T cell, this B cell will only make the same B cell. Same B cell, same B cell. So there is no memory, there is no differentiation, and there is no class switching here. However, there is activation, so it's active. So it will fight an infection. It will fight this current infection. However, it will not be able to do other things. This will come in handy later also. As far as antigen processing and presentation. So what is the cell that is going to be the first cell to interact with these antigens? What are the cells that first come to an immune response? Those are basically those are your foot soldiers, the ones that are ready. They're the ones that are waiting all the time. They're running around and looking for something that is foreign. That's going to be your phagocytes. Okay, your phagocytes. Your phagocytes, they're going to take these items and they're going to digest them and they're going to present them either as MHC class 1, MHC class 2. The main difference between them is the route. Usually MHC class 1 is associated with endogenous antigens, whereas exogenous antigens are often processed by an MHC class 2. This is very important, okay? So this is an exogenous route, MHC class 2, whereas class 1 is an endogenous route. Those are going to interact with CD8. Those are going to interact with CD4 cells. A brief summary. Cells for the antigens for the HLA are encoded on chromosome 6. There are different classes with MHC class 1, MHC class 2. The HLAs that encode for MHC class 1 are different than the ones that encode for MHC class 2. We have HLA-A, HLA-B, HLA-C, HLA-E, D, E, F, all encode for MHC class 1. When it comes to MHC class 2, we have HLA, a lot of them, DR, DP, DQ. You will come across all these things. These are called major histocompatibility complexes. Major histocompatibility complexes are important for antigen presentation. They're also important when it comes to matching tissue. When you're doing transplant, you know you need to match these MHC molecules because they are going to be the molecules that present antigens to these lymphocytes. And when they're not matched, they are going to present the wrong antigens to the lymphocytes, and they're going to cause an immune response, which is not something that you want. So you want to try to, and they're highly polymorphic, meaning they're different. Not everyone has the same HLAs, and that's why you want to try to match as much as possible. There is something called the minor histocompatibility complex. These are items we have found that when they're not matching, it doesn't necessarily lead to rejection. However, they are different among people. Example would be the Y chromosome. Our immune cells interact with receptors. Now, these receptors are found on immune cells, and we have cells of the innate immune response, and we have cells of the adaptive immune response. For the most part, your innate immune response receptors are basically pre-formed. We already have them. They're already circulating, and they're general. They're made against so many different foreign shapes, and they recognize certain molecular patterns on the surface of a pathogen, and they interact with it. And and examples would be PRRs, pattern recognition receptors. Pattern recognition receptors. Let's say they recognize a certain something on the surface of E. coli. They interact with it, and so on. We have also toll-like receptors, TLRs, and there are about 13 TLRs that we know now. And these also are general, and they're going to interact with certain molecular patterns. The most common one that we come across would be TLR4, which will bind to LPS. But that is not the only thing it binds to. So you will find that, okay, you have TLR2 or TLR3 or 6. They might bind to viral infections, they might bind to bacterial infections, and so on. So these are general. Another example would be complement receptors. They're complement receptor 1, complement receptor 2, 3, 4, and they're found on different immune cells, and they also interact with specific things. And also we have the receptor for the FC portion of an immunoglobulin. That is, all immune cells, they're going to have a receptor that is going to be specific for either IgG or IgE in the case of basophils and mast cells, and that is going to be important in triggering hypersensitivity reactions in the case of IgE. But for the most part, IgG and IgM, they interact with that immunoglobulin or that antibody, and they clear it, and they cause the opsonization as they activate complement and interact with that. Those are all general. They are not specific. They're not generated. We don't make them. We already have them. Now, we have, on the other hand, your generated receptors. And your generated receptors, those are your T cell receptor, TCR, and your B cell receptor, BCR. And those are the targeted receptors. Those are the specific receptors. And one thing you need to know about these receptors is there's only one of them per shape. There are not many, right? There's only one TCR for one shape. These receptors, you also need to know that they are shape driven, meaning they don't know if that shape they're interacting with is foreign or self. They just know that we are going to interact with this shape. Now, if they interact with our own cells, then we have autoimmune disease. So what do we do to prevent this from happening? Well, as we generate these receptors, they go through a process of selection, and eventually they go through a process of tolerance. Which part of this process is negative selection, where cells that are basically going to interact and bind tightly to our own cells, we eliminate them, we apoptose them, we delete them, we don't want them. So there's only one TCR per shape, there's only one BCR per shape, and that's it. But then, how do we have so many of them? How are we able to generate so many, such a huge diversity that is going to be able to interact with anything that we encounter? And we are talking about an unlimited, basically, number of these T cell receptors and B cell receptor, so to speak. What are these receptors? How do we make them? We have to go through this today. These receptors are here on the left side. This is your B cell receptor, and this is your T cell receptor. And your T cell receptor is a heterodimer, okay? And that heterodimer is composed of a two transmembrane polypeptide, here and here. This is alpha and this is beta, alpha and beta. So it's made of a alpha beta heterodimer. Now the chains are covalently linked by disulfide bonds. Disulfide bonds, and consist of three regions. We have the extracellular region here, we have the transmembrane research area here, and we have the intracellular area here. And this intracellular area is usually called the zeta chain. And that zeta chain is part of the CD3 complex that is going to stabilize this whole receptor. And it's going to transduce the signal all the way into the nucleus of that cell, and it's going to cause the upregulation of certain expression transcription factors. Always, a T cell is going to have a TCR. A T lymphocyte always is going to have a TCR, and that TCR is always going to have a CD3. So every single T cell, whether it's a CD4 or a CD8 T cell, it is going to have on its surface a CD3 molecule. Okay, we use that as a marker for T cells. Now, on the other hand, you have your B cell receptors, and that is also known as an immunoglobulin. And that immunoglobulin is made of a heterodimer. A heavy chain has got two identical heavy chains and two identical light chains. And this heterodimer is also composed of an extracellular region and intracellular region, and it is composed of this molecule here. It's called the CD79, and that is what's going to transmit and transduce the signal all the way inside to the nucleus and upregulate certain transcription factors that are going to activate that cell. Now, on the right side, we have some receptors. These are not generated receptors. These are preformed receptors, and those are the receptors that are going to bind to the FC portion, this portion here of an immunoglobulin. This is a summary of the interactions between a T helper cell and an antigen presenting cell, and also on the right side, a T cytotoxic cell and any target cell. And always remember, let's start with the cytotoxic T cell. Always remember, every T cell that is cytotoxic needs to be presented with an antigen. But that antigen can be presented with MHC class 1, which this target cell can be any cell of your body. And that is important. We want your cytotoxic T cells to interact with any cell that is infected with a viral infection, with an intracellular infection, or any cell that is abnormal, such as a tumor cell. And we're going to have your MHC class 1 is going to interact with your T cell receptor, and your T cell receptor is stabilized by the CD3 molecule here. Now, certain other adhesion factors like CD2 binding to LFA3 and all of that is going to be important to keep the cells bound together. Now, if you look at the left side here, we have an antigen presenting cell, which can be a B cell, because B cells are also antigen presenting cell, but can be a macrophage, can be a microglial cell, can be a Kupffer cell, a dendritic cell, and so on. It even can be an astrocyte, because astrocytes are weak antigen presenting cells. In this case, it's an MHC class 2 that is going to interact with your T cell receptor, stabilized by CD3. But this molecule here, a T helper cell, displays on its surface not CD8, it displays on its surface a CD4 molecule. Now, there are other additional molecules that are required to activate a T cell. and that is a co-stimulatory signal. And that co-stimulatory signal is called the CD28 on the surface of the helper cell. It becomes expressed and it binds to a B7 molecule. And once this binding happens, so first we need the signal, a peptide presented to this T cell, and then we need a second signal to activate that cell, which is the CD28 here binding to the B7. And that will result in the activation of that T cell. Now we are going to go through the process by which we make these T cell and B cell receptors. And that is actually the going through the development of these lymphocytes. And this is very important. And this is a schematic. It is found in Abbas, the immunology book. And basically, it takes you through the process, both B cells and T cells. Obviously, it is different between them, but general idea would be we always start with the stem cell. The stem cell is a hematopoietic stem cell. Okay, it's a pluripotent cell, which means that stem cell can become anything. It can become any blood cells. It's a hematopoietic stem cell. Now, that cell, whether it's a B cell, whether it's going to be a B cell or a T cell, it goes through the same process. So if it's a B cell, it becomes a progenitor cell once we start assembling that immunoglobulin on its surface. Same thing if it's a T cell, it becomes a pro-T cell once we start assembling that T cell receptor on its surface. But for the most part, we go from a stem cell, which is a pluripotent hematopoietic stem cell, which becomes a progenitor lymphocyte. And that progenitor lymphocyte, it starts rearranging and making that cell receptor, be it a B cell or a T cell receptor. And then it becomes a pre-lymphocyte once this process is almost done. And when it's assembled, it becomes an immature lymphocyte. That immature lymphocyte, in the case of a T cell or a B cell, basically they go and they hide in the peripheral lymphoid organs and they wait to be activated. They get activated by interaction with some kind of a protein, some kind of an antigen, I'm sorry. We're going to go into this process in a lot more detail because that is basically the essence for hypersensitivity and autoimmune disease that occurs. So lymphocytes, they come from a hematopoietic stem cells, HSCs. And under the influence of certain transcription factors, they can become, so they go from a hematopoietic stem cell into a common lymphoid progenitor. And that lymphoid progenitor becomes either a pro-B cell or it can become a T cell, a precursor for a T cell. The pro-B cell will become a pre-B cell as it starts making that immunoglobulin, and then it's gonna become, depending on where it is, if it goes to the follicular areas of the secondary lymphoid organs, it becomes a follicular B cell. And those follicular B cells are B cells that will only be activated when they interact with a T cell. So they need the help of a T cell in order to activate. They hang around in areas where there are other T cells. Now, B cells can also go to the marginal zone area of the secondary lymphoid organs. These marginal zone B cells are B cells that are able to activate without the help of a T cell. Usually, these are the B cells that are going to interact with your polysaccharides. I'll show you why. Now your T cells can become a pro-T cell, which leads to a pre-T cell, which will become either an alpha-beta T cell, or in very rare occasions, like less than 5% of the cases, it becomes a gamma-delta T cells. And these gamma-delta T cells, we find them in the gut area. Your alpha-beta T cells are the alpha-beta T cells that we find them everywhere in the secondary lymphoid organs, again, waiting to be activated. How do we make that T cell receptor? Or how do we make that B cell receptor? How do we account for this unbelievable diversity? The human body, as humans, we have a certain numbers certain number of genes. Do you know how many genes we have? We probably have 20 some thousand genes, maybe 30,000. I don't know, the number escapes me. But if you combine these genes in any possible way, there is no way you can account for the unlimited number of lymphocytes that we make. And remember, every lymphocyte is shape driven. It will interact with only one shape. Think of it as a key that will only unlock only one lock, right? Now, in order to account for this diversity, the genes that encode for these receptors are basically gene segments. And these gene segments are found on different chromosomes. And these gene segments can be arranged in different combinations. And if you look here on the top of the screen, we have the structure of an immunoglobulin. And on the bottom, we have the structure of a T cell receptor. So the structure of the immunoglobulin is also the B cell receptor. And your B cell receptor is made of two identical heavy chains and two identical light chains. And then the T cell receptor is made of an alpha chain and a beta chain, and an alpha chain. The beta chain is analogous to the heavy chain. It has a similar structure. And so does the alpha chain in the case of the light chain. The light chain of the immunoglobulin also is very similar in structure to the alpha chain of a T cell receptor. The heavy chain is made of different gene segments. And these are the variable region, the diversity region, and the joining segment. And there's so many different variable D and J. We recombine them in so many different, like V1 comes to next to let's say a D6 come next to a J2, next to a V20. And now the light chain is very similar, but it does not have a diversity area. Same thing with the TCR. It has a V, a D, and a J for the beta chain, but the alpha chain, just like the light chain, it only has a variable and a joining segment. Now they all are followed by a constant region. And usually that first constant region we hit is going to be called the mu region. And that's why when you're making an immunoglobulin, you hit the mu, and that is why you first make an IgM, right? Just by the reason where it lies. It just happens that it's the first constant region that we are going to hit. So how does rearrangement occur? Now, before I talk about this, are you fine? Is the material I am covering right now, is it something that is very simple to you? Something that you already know? So this is an important concept here. The concept of allelic exclusion. The concept of allelic exclusion is very important because as you make a lymphocyte, there are different alleles that make the T cell receptor or the B cell receptor. And in the case of the B cell receptor, I'll use that as an example. You get alleles from the mother and you get alleles from the father. But if both alleles are expressed, then we will have two different lymphocytes, which we can't. For every shape, there's only one lymphocyte. And the cells, the alleles come only from one of the parents, either the mother or the father. And that is how we prevent having multiple lymphocytes for one shape. And the way we do that, accomplish that, is by a concept called allelic exclusion. And you have to follow me here. So as we start rearranging making the B cell receptor, we have to make the heavy chain first, and then we make the light chain. And then we put the heavy chain and the light chain together, and we have our immunoglobulin, our B cell receptor. So we start rearranging the heavy chain, and that happens in the pro-B cell. And we start arranging the VDJ part of the heavy chain. Now, the genes for the heavy chain are found on chromosome 14. So we take chromosome 14 from only one of the parents, either the mother or the dad. It really doesn't matter. And we start rearranging. And we start by rearranging the D and J. and that will be the pro-B cell part. And once that is complete, we go to we complete the pro-B cell by finishing the VDJ part. Now, we start with one of the parents. If this process is successful, we go on to make the light chain. If this process is not successful, we try the chromosome 14 from the other parent, right? And if this is successful, we go and we try to make the B cell light chain. If this is not successful here, then we apoptose the cell. So you have two chances of making a heavy chain. A chance 1 from one parent and chance 2 from the other parent. If both chances are not successful, we apoptose. Now, if this chance is successful, we go on to make the light chain. And the genes for the light chain, we have two different light chains, unfortunately, to make things complicated. We have a lambda and a kappa. We can only do one of them, but then the genes for the kappa are found on chromosome 2, and the genes for the lambda are found on chromosome 22, and we only need one of them. So we always, for the most part, usually we start with the kappa, and your most BCRs are kappa light chain. And we go, just like the heavy chain, we start the rearrangement process on one chromosome from one of the parents. So let's say we start the kappa on the mother this time. And we start rearranging. If this is successful, we put it next to the heavy chain that we already made, and we have an immunoglobulin. If it's not successful, we go to the kappa of the other parent, and we start working on it, and we make an immunoglobulin. If this is not successful, then we try the lambda on chromosome 22 from, let's say, the mother, and we try to make it. If this is successful, we put it next to the heavy chain and we make an immunoglobulin. If this is not successful, we go to the lambda on the other parent, chromosome 22, and then we try to make it. If this is not successful, then we apoptose the cell. If this is successful, we make an immunoglobulin. So basically, in a nutshell, the summary of this whole thing, we have 2 chances to make a heavy chain, we have 4 chances to make a light chain, and we have a total of 8 chances to make an immunoglobulin receptor, a B cell receptor. You need to know this. This is a common question that you will encounter.